Welcome to another UK Freshwater Quality Research Program webinar. We do this every month, so probably you have been here before, but it's always worth talking a bit about who we are and what we are doing. So we are a NARC and DEFRA funded research program. We are working to basically strengthen our understanding of and knowledge about sources and behavior of different pollutants within the UK river systems. We are working to increase the knowledge and understanding of how these pollutants and chemicals are impacting the river systems. Ultimately, our aim is to inform policy and practices to improve the ecological health of UK rivers. So we are a program team, and there are 5 very impactful projects within the team. There are 5 impactful projects working under the program, and there is a broader program team that is overseeing execution of this projects. I am part of the program team. Pardon, I forgot to introduce myself at the beginning. So I am Farhana Naz. I am within the program team as a program support, and today my colleague Richard Grayson will also help us with this webinar. So as you can see, there are the projects listed on this slide. We'll move on to the next slide. So you have different ways that you can connect with us. As I mentioned, we do this every month, these webinars, and they are recorded. So if you scan the QR code, you will be taken to our website where you will find all the recordings of our previous webinars, and you can catch up with them. You can also email us at freshwaterquality@leeds.ac.uk if you have any questions or know more about the program. We also have social media presence. So before I hand over to our speaker for this webinar, Elitsa, I just want to quickly mention the upcoming events we have. The next one, we are not going to have one in August because of the holiday season, but we have the next one upcoming on 18th of September, and then the one after that will be on 23rd of October. I think now I'll request our speaker to come up and talk about her work. Hand over to you, Elitsa.
So my name is Elitsa. I'm a PhD student at the University of Exeter, and I work on the environmental dimensions of antimicrobial resistance and its implications to public health. Today I will talk about antimicrobial resistance specifically in freshwater environments, and I will introduce the PUSTIC study with some preliminary findings as this work is still ongoing. First, let me explain that antimicrobial resistance is an umbrella term which describes the evolution of resistance in microbial pathogens, such as bacteria, to antimicrobial drugs like antibiotics. But when I talk about antimicrobial resistance today, I won't go into any of the rest of the pathogen types or antimicrobial drug types. I will specifically be talking about bacterial resistance to antibiotics. So antimicrobial resistance is a natural process where microbes evolve or change over time to no longer respond to medicines such as antibiotics, which we use to treat them. So this makes recovery from these infections particularly challenging. Having less effective drugs obviously means longer illnesses, longer hospital stays, and higher risk of patient death. And in fact, today, antimicrobial resistance is considered one of the top global health threats, as it has been estimated that each year about 5 million people are killed either directly or indirectly by resistant bacteria globally. And if we do nothing to stop this, in about 25 years or so, we can expect to see bacterial infections once again becoming the leading cause of death, with 1 person dying every 3 seconds. So what can we do? When we talk about antimicrobial resistance interventions, we typically focus on antimicrobial resistance in the clinic or in veterinary medicine, which makes sense because this is where the majority of antibiotics are used for. But the problem is that if we hope to somewhat control the spread and maintenance of AMR or antimicrobial resistance, we need to understand that this is not a human and animal health issue. This is a planetary health issue, and So today I'd like to shift the focus onto one of the more overlooked antimicrobial resistance domains, which is the natural environment. And obviously I'll have a specific focus on natural waters. Why natural waters? Well, these environments bring all the necessary components together for the emergence and maintenance and spread of antimicrobial resistance. So first we have a diverse load of environmental bacteria, which not necessarily pose a direct threat to the public. Some can, of course, but the reason environmental bacteria are particularly interesting is because they're actually a rich source of antimicrobial resistance mechanism. And in fact, some important mechanisms, such as beta-lactam resistance, are considered to have originated from environmental bacteria and then subsequently spread onto clinical pathogens via horizontal gene transfer, which is the ability bacteria have to share genes between both related and unrelated species. And essentially what this means is that once a resistance mechanism has emerged, this can then quickly spread, provided there is selection for that gene. So the point I'm trying to make here is that while environmental bacteria are not necessarily a direct threat to our health, they can be a significant source for jumpstarting the evolution of resistance for human and animal pathogens. And human and animal pathogens are also present in these environments as we continuously release waste, such as effluent, raw sewage outflows, and manure runoff. So with these sources of waste, we're enriching continuously these environments with human and animal bacteria, which will include some pathogens or disease-causing bacteria, and some of these will already carry resistance mechanisms. Alongside microbes, our waste also brings various antimicrobial resistance driving pollutants, such as antibiotics, for example, which a large part of antibiotics can be excreted unmetabolized through our urine and feces. But antibiotics are not the only driver for antimicrobial resistance evolution, even though they're the primary one. Research, including in our own lab, has shown that some non-antibiotic drugs, such as ibuprofen, for example, but also antimicrobial disinfectants, some pesticides and herbicides, and metal pollution can also bring advantages for bacteria carrying resistance mechanisms. And this may then be further contributing to the emergence, maintenance, and spread of antimicrobial resistance in the environment. So basically, we consider natural environments to be both a hotspot and a reservoir for antimicrobial resistance emergence and spread, because these environments naturally harbor diverse environmental bacteria, because they receive, through our waste, human and animal bacteria, as well as antimicrobial driving pollutants, such as antimicrobial drugs, but also disinfectants or pesticides. So this is what happens in theory. We can show evidence of these processes actually occurring in the lab. We can also trace antimicrobial resistant genes from clinical pathogens to environmental bacteria through DNA studies. But what is the actual evidence that from the field that environment to human transmission of antimicrobial resistance is something that we need to be concerned about? So what we do know is that the levels of resistance typically increase downstream of sources of pollution, so something like an effluent discharge point or a farm, for example, relative to upstream samples. We also know that freshwater swimming has already been identified as an independent risk factor for acquiring drug-resistant urinary tract infections from a study in Norway, and that overall, during the summer months, freshwater swimming is considered to account for about 6% of antimicrobial resistance acquisition in the community, and this comes from a study in the Netherlands. But when it comes to studies directly addressing environment to human transmission of antimicrobial resistance, the literature is even more limited. So the first ever study to directly explore the link between water-based recreation and antimicrobial resistance in terms of gut colonization was the beach bum study done by my now supervisor, Anne Leonard. She chose to work with frequent surfers because surfers can be considered a high-risk group. And this is because they tend to spend a lot of time in the water, but also they're more likely to ingest more water unexpectedly because they tend to fall unexpectedly, and therefore they're more likely to swallow more water. So assuming that the water at the time is containing any pathogens, this then increases the likelihood of this group of water users to then ingest any pathogens that may be present at the time. So the idea basically here was that if bacteria survive ingestion, they could colonize the gut. And since surfers seem to be at more risk of ingesting lots of water, and then therefore we can expect to see that there is a higher prevalence of gut colonization with antibiotic-resistant bacteria in their gut, which is what Anne was looking for in this study. So she was screening fecal samples. She was actually screening swabs for the presence or absence of antibiotic-resistant E. coli or E. coli resistant to third-generation cephalosporins. And this organism serves as the World Health Organization One Health surveillance target organism. So it's used to screen the levels of AMR in humans, animals, and the environment as well. And she found that the likelihood of surfers testing positive for third-generation cephalosporin-resistant E. coli was 3 times greater relative to people who don't go into the water. So she found exactly what she was expecting for, basically. And then a couple of years ago, there was a second study which came out exploring the link between water exposure and antimicrobial resistant gut colonization. But this time the focus was on coastal swimmers. And a little surprisingly here, the study didn't find the same trend as Anne did with her surfers. So here, coastal swimmers were found to be actually at a reduced risk of carrying resistant Enterobacterialis relative to the control population of non-swimmers. And Enterobacterialis here is just the order level, which means that the researchers here did not restrict their surveillance to E. coli specifically. So there are several proposed explanations for why this may have happened. So first, they used different methods, though I personally don't see why this should impact the outcome as much, at least. The other suggestion was that records of water quality in Ireland show that the water was much cleaner relative to the water in England at the time when Anne was doing the beach bum study. But probably more importantly, at least this is my opinion, the focus here was on a different group of water user. So the majority of participants here were swimmers, not surfers, which, as I mentioned before, were selected in the beach bum study as a high-risk group. And in fact, the researchers did, in this second study, they did note in their paper that a higher proportion of people who engaged in water sports involving boards, such as surfers, were found to carry antibiotic-resistant Enterobacterialis relative to other water users. So overall, what this study indicates is basically that perhaps swimming along the Irish coast is just not a risk factor for being a gut carrier of antimicrobial-resistant E. coli, which I personally think is great, especially if you're a swimmer in Ireland. But I wanted to challenge this exact same question for the exact same group of water users, but I wanted to do it in a different natural environment. So with the PUSTIC study, I wanted to test whether wild swimmers, specifically those visiting natural inland waters, so anything like rivers, lakes, dams, etc., across the UK were at greater risk of carrying antibiotic-resistant E. coli relative to people who don't go into the water. And the reason why I wanted to do this work is because wild swimming, including in freshwater environments, is a growing trend, not just in the UK, but also globally. And importantly, wild swimmers visiting rivers and freshwater environments specifically can be exposed to higher levels of fecal pollution from effluent, sewage outflows, and manure runoff, because unlike coastal waters, in freshwater environments, the capacity for pollution dilution is much reduced. And of course, still there's very few swimming locations along inland bathing waters that are actually being monitored for indicator fecal bacteria. And with the exception of bathing sites in Scotland, there's no other country across the UK that I know of, or globally, that are monitoring any bathing sites for specifically antimicrobial resistance levels at all. And this leaves wild swimmers completely uninformed about what risk they might be exposed to. So to address this question, I started recruiting participants over the course of last summer for the post-EEC study. I was hoping to get 150 fecal samples from wild swimmers and 150 fecal samples from non-swimmers over the course of the bathing season. I was then able to screen these samples for the presence of third-generation cephalosporin-resistant E. coli, which, as I mentioned before, serves as the One Health surveillance target organism. This bacteria is also on the WHO's list of priority pathogens because of its high disease burden, limited treatment options, and high levels of transmissibility. So the eligibility criteria to take part in the post-EEC study were very strict for both swimmers and non-swimmers, and this was to ensure that only healthy individuals with no or minimal risk factors for gut carriage of antimicrobial resistant E. coli were able to take part. So this meant that none of our participants had known chronic conditions, no participants had traveled outside the EU in the past 6 months, none of them had an overnight stay in a hospital or a healthcare facility in the past 6 months, none of them had taken antibiotics, or lived with someone who had taken antibiotics, again in the past 6 months. And there was a huge interest in this study, with over 1,500 participants expressing interest to take part. Of these, about half were eligible, and another half actually returned their kits. So we ended up with 347 participants returning their samples. Of these, 41 participants were excluded. For some circumstances had changed. For example, they had to take antibiotics, or some didn't meet the eligibility criteria. So for example, they swam in the sea rather than a river or a lake. And this left a final count of 306 participants, 138 of which were swimmers and 168 of which were non-swimmers. So here you can see that the distribution of participants is fairly well distributed. I mean, it could be improved, obviously, but I don't think I can ask any more from participants. There are some clusters, for example, around London that you can see. So these maps are based on the first parts of the participant postcode, so they're an approximate location, and on the left in blue is the map showing the distribution of swimmers, and on the right in yellow is the distribution of non-swimmers. And here I wanted to include also a map of all the reported swimming locations. So for each X here, that indicates a swimming location. So it's not a swimmer. We had 138 swimmers, which reported a total of 258 swimming locations. So one person can be visiting multiple locations. And I wanted to include this slide here just to show the extent of wild swimming happening across the country, and to reiterate that this is not just an isolated community somewhere confined to very few popular swimming locations. Wild swimming is happening all over the UK. Swimmers reported visiting 159 natural inland water bodies, the majority of which were rivers and lakes, but people also reported swimming in reservoirs, estuaries, ponds, pools, quarry, lochs, waterfalls, although I'm not sure how you swim in a waterfall, but there we have it. The most visited river was the Thames, with 27 participants reporting to swim there. And this was followed by the rivers Dart, Wharfe, Nene, and Lake Windermere. The majority of wild swimmers in the study reported swimming in the past week prior to submitting their sample. The majority also reported having had over 7 head immersive swimming sessions in the past month, and over 25 head immersive sessions in the past 6 months. So the typical time spent in the water for most swimmers was between 15 minutes to an hour. And overall, from these, you can see that our swimmers were individuals with regular exposure to natural inland waters. They weren't bathing every now and then. They were frequent bathers. So now I'm going to go over some of the demographic characteristics of our participant population. So the bars in blue here represent the swimmers, and the bars in yellow represent our control group of non-swimmers. As you can see, the majority of swimmers in our study were mostly female. They were between the ages of 45 and 65, while in the control group, that was predominantly over the age of 55. Participants were predominantly white, with at least one, predominantly with at least one higher education qualification. In addition to demographics, we also asked about a range of risk factors which could potentially increase the risk of carriage of resistant E. coli. So first we asked about occupation in terms of antimicrobial resistance, healthcare workers and people working with animals, as well as those people living with either of these professionals can be at higher risk of being carriers of resistant E. coli. And we did find that our swimmers were more likely to be healthcare professionals or to live with one compared to the control group of non-swimmers. But this wasn't associated with antibiotic-resistant E. coli, which means that being a healthcare professional, at least in this particular cohort, did not translated into testing positive more often than what you would expect by random chance. We also asked about travel abroad, living with cats and dogs, or having water from a private source, but there were no major differences between any of these risk factors between our swimmers and non-swimmers. And participants were also asked about diet, in particular whether they consumed beef, pork, lamb, chicken, shellfish, fish, and salad, which are again non-risk factors for antimicrobial resistance gut carriage. And the only difference here that we could find was that our swimmers were actually less likely to consume beef and pork. But again, none of these were associated with outcome. We also asked about alcohol consumption and consuming pro and prebiotics, but there were no major differences in the observed between the two groups. So the potential confounders here associated with being a swimmer were sex, age, occupation, beef and pork consumption. But again, none of these were associated with outcome, which means that none of these increased or decreased the risk of being a carrier of resistant E. coli in this particular cohort. And despite this, we found that overall the prevalence of third-generation cephalosporin resistant E. coli was similar to the prevalence of found in the Surfer study from the beach bum study. Now, I won't be able to share the exact figure with you, but they're more similar to the beach bum study than the Irish coastal swimmers. And the reason why I'm not sharing the exact numbers is because we are in the process of publishing this. So I'm just going to have to keep you in suspense. But we also found that wild swimmers were more likely to carry BLAST-TXM gene, which is the most important antimicrobial resistance mechanism to beta-lactam in Enterobacteriaceae, which again is similar, although a little bit higher than what we see in the beach bum study. Although I'm not sure if I reported that data in my previous slide, you'll have to let me know. So we also found that wild swimmers actually carried more diverse phylotype of antimicrobial resistant E. coli, and they were more likely to include phylotypes which are frequently reported as a cause of for extraintestinal infection, though this could be just due to having a higher number of swimmers testing positive relative to controls, of course. And interestingly, in addition to exposure to natural waters, carriage to resistant E. coli was also associated with alcohol consumption, particularly with consuming alcohol over the recommended 14 units per week, though the number of participants who did report consuming that much were actually very small, so this finding should be taken with caution. There is also an indication that geographic location might be important here, though again, the population sample was not large enough for this type of analysis, but just to show you what I mean here, I'm going to show you this map where you can kind of see that there is a disproportionately higher number of positive swimmers in the south of England relative to the north, which is not something that we see in our control group. So I should explain what is going on here first. So I've only included a map of England here because less than 10% of all participants came from elsewhere. So from Scotland and Wales, there was no participants who came from Northern Ireland and none from Ireland either. So here we have two heat maps in blue or gray-ish. The one on the top is showing the total count of swimmers, and the one on the bottom is showing the total count of non-swimmers. And these are divided by each of the 9 main regions in England. On the right-hand side here, in red, we have the percentage of positive swimmers for each region on top, and the percentage of positive non-swimmers on the bottom. So the color, where the color is white, this represents 0 participants within the region, and the darker the color in whichever category, the more participants there are in that region. So what these maps are showing is that essentially the majority of positive swimmers came from the south. At first, I assumed this is probably skewed from the cluster of participants which came from London, but because this is an area with generally higher prevalence of antimicrobial resistance in the general population compared to the rest of the country. But as you can see from these maps, this doesn't seem to be the case here. And while living in London, might not be the reason why we're seeing these numbers. Swimming in the River Thames actually gave a kind of a better clue. So River Thames swimmers represent just under 20% of all the swimming participants, but they accounted for 50% of all the positive cases. So I'm just going to repeat that again. The swimmers that go swimming in the River Thames represent under 20% of all swimming participants, but they account for 50% of all the positive cases that we saw. So, out of all the rest of the wild swimmers that lived in the same areas as River Thames swimmers, but who didn't swim in the Thames, they swam elsewhere in other inland bathing water locations. None of those tested positive. And this indicates that there might be an elevated risk that is specific to the River Thames. And here, I should also note that swimmers in the south, which tested positive, were not restricted only to the River Thames. This is just where the majority of positive swimmers came from. And before I wrap this up, I'd like to remind everyone that while we are seeing a higher proportion of positive samples from wild swimmers compared to controls, we can't claim that wild swimming is the cause. After all, the PUSTIC study was just an observational study, and since we are not able to screen participants for their colonization status before swimming, we can't with certainty say that swimming is why these people have acquired these E. coli in their guts. But by excluding people who had been exposed to other environments with known sources of antimicrobial resistance contamination, we hopefully were able to minimize the effects of these. Right, so what's next with the PUSTIC study? There's a lot of sequencing in bioinformatics in my future. We have just completed metagenomic sequencing of all of our samples, which is really exciting. This will allow us to look at the entire resistome within each sample, which would provide a better overview of the AMR gene diversity within the samples than simply looking at one target organism. Amplicon sequencing would help us understand the microbial diversity within our samples. We would also be quantify specific genes using qPCR for high sensitivity. And we'll also be using whole genome sequencing to understand what other resistance mechanisms might be in our already collected positive isolates. And before I take any questions, I would like to mention quickly that at the moment, I'm also running a second study. So this study does not look specifically at any specific pathogens at all, but or antimicrobial resistance. It more broadly explores whether swimming is associated with a high risk of developing symptoms of infection compared to people who don't swim. So here, anyone over the age of 16 living in the UK can take part, and taking part means simply completing a short questionnaire, which you can access by scanning the QR code on the screen. If you can, I would really encourage you to take part, especially if you're not a swimmer, meaning that you have not been swimming in any pools, rivers, lakes, the sea in the past 2 weeks. And this is just because finding non-swimmers for this study is proving to be quite challenging. So I would really appreciate your help either as study participants, or even if you simply share this information about the study with your social networks. And I'll end this with thanks to everyone who helped me make this work happen, including my supervisory team, but also to all of the participants who took part, and to everyone who shared this study online, because it's definitely been a team effort. And thank you all for listening.